Beware of herpes.
Herpes is a virus that is characterized with a large double-stranded DNA genome in a glycoprotein-bearing lipid bilayer envelope and four-layered structure.
There are two types of herpes, genital herpes and oral herpes. Oral herpes is an infection of the lips, mouth, or gums due to the herpes simplex virus. It causes small, painful blisters commonly called sores or fever blisters. Genital herpes is a common and highly contagious infection usually spread through sexual intercourse. This infection is usually caused by the herpes simplex virus 2 or the herpes simplex virus 1. The virus is usually responsible for cold sores.
T cells are used to immune the cell that is infected with the herpes virus. When the antigen and the MHC molecule appears to notify the T cell to help it. Then the herpes virus affects the cell by entering through the cell and leaving the strands of its own DNA throughout the whole cell. But as soon as it penetrates the nucleus, it infects the whole cell. Later, it attaches itself to the molecule. This helps the virus produce more of the herpes virus. The worst case is when the cell bursts and it causes the inflammation, pain, and spots with live virus.
Herpes multiplies by transcription, genome replication, and capsid. It occurs in the nucleus of the host cell.
Transcription is the first step of the gene expression, in which a particular segment of the DNA is copied into RNA by the enzyme. RNA polymerase, both RNA and DNA are nucleic acids, which use base pairs of nucleotides as a complementary language that can be converted back and forth from DNA to RNA by the action of the correct enzymes during transcription. A DNA sequence is read by an RNA polymerase, which produces a complementary RNA strand.
Genome replication is the process of producing two identical copies from the original DNA molecule. This occurs in all living organisms and is the basis for biological inheritance. DNA is composed of two strands, and each strand of the original DNA molecule serves as a template for the production of the complementary strand. Cellular proofreading and error checking mechanisms ensure near perfect DNA replication. DNA replication begins at specific locations or origins of replication in the genome, unwinding of DNA at the origin, and synthesis of new strands result in replication forks growing bidirectly from the organ.
Capsid is the protein shell of a virus, and it consists of several subunits made of proteins called proteiomers. The three-dimensional subunits may or may not be correspond to individual proteins, are called capsomeres. The capsid encloses the genetic material of the virus. Capsids are broadly classified according to their structure. The majority of viruses have capsids with